Right, last class we spoke about mainly osmosis, right? Then the different types of solutions, then what happens to the cell when it is placed in each of these solutions. All these things we had discussed. Then we also took example of a grape and raisin and discussed the scenarios, different scenarios of what will happen, etc. So last class we just did osmosis in general and a little bit about plasmolysis, right? So just to explain this part, I would go back one slide so that, yeah, where we were, we started. Yes. So, yes, we discussed plasmolysis and then, okay. Yes, we then came to cell wall. We discussed where it is, location, etc., and its function. Important here it is. What is plasmolysis? Right. So we discussed till here, and I was supposed to tell you or discuss in detail about wall pressure as to why is wall pressure important or how does the cell wall help or how is wall pressure one of its main functions. This is what I wanted to discuss. So we'll start from here. Yeah. Okay. Right.
Means
So this,
everybody is not.
tell me. Everybody is not here yet, right? Yeah, I know. But if I wait, then I have to stop also at dot 4:30 because some of you have another class, right? Okay, I'll just put a message saying we started. Let's see. Right. Okay. Yes, you all can switch on your cameras. I think this batch is
Yeah,
not
I'm just eating food.
okay. Okay. Okay, I think everybody is here except for Neil, isn't it? Is everybody here? Okay, I'll ask Neil where is he? Okay, yes, sure, Aliza. No worries. You can switch on in 5 minutes. Okay. Right, so I just discussed what we did last class. For those who just joined in, I was just doing a recap that we covered osmosis last class and we covered the different type of solutions based on the solute concentration. Then we discussed what happens to a cell when it is placed in these solutions. We also discussed, we took some case-based scenarios like what happens if you put a grape in these solutions or what happens when you put a raisin in these solutions, etc. We discussed all that and we spoke a little bit about cell wall. Right, we stopped at plasmolysis, which was one of the conditions which occur in plant cells where the cell contents shrink away from the wall. Right, so we discussed cell wall, where it is present, location, what is it made up of, and its functions. So one of the functions of the cell wall was wall pressure, and I had said that I would explain it in the next class. So we are starting from there. Okay. You don't need to draw this. I'll tell you when you need to take it down. This is just for you to understand these two terms because you might come across them in some questions. Okay, wall pressure and turgor pressure. These two terms, if you come across, you should know what they mean. For that reason, only this slide is included. Right. Okay. Okay. So what happens? You take an example of a balloon. Okay. For example, you have a balloon which is not inflated, right? Deflated balloon basically. Okay. It looks something like this. It is shapeless. You can, you can pull it, you can stretch it, you can make a weird shape out of it basically, right? Or it's going to just move around as it's on its own without having any form of stable structure basically. But what happens when you inflate the balloon, right? When you put in sufficient air in the balloon, you inflated it, right? And then you try to change the shape of the balloon, it does not really move much, right? The shape does not get altered that extent as in the case here. Okay. Same thing happens in cells. In order for the cells to maintain their structure, so basically if your plant cell is something like this, right? In order for the plant cell to stay in this shape and not become some random weird shape, you have wall pressure and turgor pressure. Basically, wall pressure is the pressure which the cell wall exerts on the cell contents, whatever fluid and organelles are there inside, right? It is the pressure which the cell wall will exert. Okay, exerted by cell wall. You can write this, that wall pressure, what I'm writing here, you can write that. Exerted by cell wall, EXER, okay, exerted. Okay, let me write it again. Okay, exerted by cell wall, right? And turgor pressure is basically the pressure which is exerted by the cell contents on the cell wall. Okay, so something like this is happening. If this is your plant cell here, right here, which we there in the bottom, the cell wall is exerting pressure like this, and the cell contents are going to be exerting pressure, okay, like this, right? So because of these two pressures being almost equal, or being on the same magnitude, the cell shape is maintained. It is maintained as a rectangle in plant cells, rectangularish, right? And in animal cells, of course, the cell wall is not there, but you do have turgor pressure being exerted even in animal cells. Right, so these two terms are important. Sometimes they ask you what is turgor pressure, so you just need to know that it is the pressure exerted by the cell contents. What are the cell contents? Basically all the organelles here, the matrix, which is nothing but the cytoplasm, which we will discuss in the next slide, right? All these things are the cell contents. Okay, matrix is nothing but the cytoplasm, right? So that's it. That's exactly what they have explained it to here. Cell contents exert turgor pressure and the cell wall would exert wall pressure. So this is why wall pressure becomes a function of the cell wall. Okay, that's how it maintains the cell structure and it maintains the cell shape. Okay. Have you taken this 2 points down?
No, just give me a few minutes.
Okay. Others, have you taken it down?
Miss Neil is still not.
Yeah, I
Yeah,
have
I'm down.
informed Neil on the group. I don't know where is he. I can't keep waiting for him, right? Okay. Okay, tell me once you all have taken it down, then we'll go ahead.
Yeah, Miss, just give me a few.
Done, ma'am.
Okay. You just have to take down these 2 points here. I'll put a star mark. You don't have to draw the diagram, just these 2 points. That wall pressure is pressure exerted by cell
Yeah,
wall,
miss. Almost done.
and turgor pressure is pressure exerted by the cell contents on the cell wall. Done?
Yeah.
What about Kushagra? Done?
Yes.
ठीक है. Right. So this is our plasmolysis diagram, which we had seen last class as well. So this is a normal cell. It is a no, I've forgotten the R here. Right, normal cell. And this is, if you place a cell in the isotonic, is the same concentration, right? So nothing happens to it. If you place it in a hypotonic solution, which means this has less solutes, so more water. So water goes in, the cell swells. So this is an example of a swollen cell, right? And when you place it in hypertonic solution, so basically this is what you need to remember, that plasmolysis will happen only when you place it in a hypertonic solution, right? Okay. This diagram again usually is not asked as you need to draw it or anything. Sometimes they just ask you to identify. They'll give you these 3 cells like this, and they'll ask you to identify which one is plasmolyzed. Okay, so you need to know how it looks, right? So this is the one which is plasmolyzed. It can be Z also. Both the spellings are correct. Okay, anything, S or Z. Okay, right. Yeah. Next, we will talk about the nucleus. So if you remember in our first class, we had discussed that every cell has
Has a cell wall, cytoplasm, and a nucleus.
a cell wall. Are you sure?
It's something that starts with cell.
Okay. Anybody else
Cell
remember?
membrane.
Okay, cell membrane. Right. So every cell has 3 features: cell membrane, nucleus, and cytoplasm. Right. This is what we had done in our first class. Correct. So
Yeah.
after this, we had discussed cell membrane, and we discussed cytoplasm, that it's the jelly-like fluid which is floating in the cell, right? Which holds everything. That is cytoplasm. We were left with nucleus, right? So here is the description of nucleus. So every cell has a nucleus, right? So remember this. This is in continuation with this. So nucleus, from here, yeah, you might come across various nicknames of cell organelles, right? Like nucleus is called the brain of the cell, or it's also called the control room of the cell. This slide is important. You can take down everything. Diagram is not required. Whenever the diagram is required, I'll tell you. Okay. Okay. It's also called a control room of the cell. Okay. And how did it get this nickname? Is because the brain is the one which will tell all the other organelles and other cell components to do, or do their particular functions. The brain is the one giving them tasks, right? So that's why the nucleus would be called the brain of the cell. Like in humans or animals, the brain is the one which controls the signals, or which ensures that messages are transferred from one part to the other. The nucleus will ensure all the cell functions are.
Miss, in this says it says contains chromosomes. Does it mean X chromosomes and Y chromosomes?
Okay, X chromosomes and Y chromosomes are present in what are called the gametes or sex cells. Okay, only they will have this. Body cells don't really have X and Y chromosomes. They have autosomes.
Uh,
Okay, they
uh,
are called autosomes.
do we need to write down the parts because are you going to be explaining the parts, right, of the nucleus?
No, no, that's not required. You need to know this. Whatever you need to know has been written here. Parts may you don't need to really know much except that it has a nuclear membrane. This is all you need to know. What else is there? So the nuclear membrane is called the nuclear envelope. Okay. Nuclear pores, they are basically just parts or methods. It's just an opening like stomata in plants. Here you have nuclear pores which allow materials to move in and out of the cell. That's the function of the pore. What does the nucleolus do right now is not required for you. What the chromatin does is there in the next slide. I'll tell you about chromatin a little bit in detail in the next slide. Right, so that's all you need to know. This is another cell organelle which we will study in a bit, right, which is attached to the nucleus. Right, so the function of pores is also given here. Chromatin is there in the next slide.
That's the endoplasmic reticulum, right?
Yes, correct. That's the endoplasmic reticulum. Okay, have you taken this down?
Or almost.
Yes,
Yes,
ma'am.
Yes, okay. What about Kushagra done? Kushagra, where is he?
yes, yes.
Done. Okay. Done, right? I'll go ahead then.
Yes, ma'am.
Okay. Right. So you need to know a little bit about chromosomes and chromatin, not much in detail. Again, because you are new to these concepts or new to these terms, it's not required in detail. But sometimes they ask you questions like, what is chromatin? Or they might ask you what is chromosome. So for that reason, I've just included the basic difference between the two. Basically, when a cell is not dividing, as you would be knowing, right? Every cell at some point or the other it divides to give rise to two more cells, right? Okay, this is how the cell undergoes continuous replication, or it maintains its species, or basically this is the way the cell continues to survive, by undergoing cell division. Okay, so when a cell is not dividing, basically when this process is not happening, the chromosomes are present as thin fiber like threads. Okay, they are literally like this, and they are called as chromatin material. Whereas when the cell is dividing, which means that this process is going to occur, this thread like structure gets condensed into structures which look like this. Okay, and these structures are basically what are called chromosomes. Right, so once you write this, you can write all of this.
Miss, just give
You
me.
can. Yes, yes, I'm waiting. I'll just tell you one more point which you can write. So you can write that this is chromatin, okay, and this is chromosome. Okay, it forms structures like this, which are called chromosomes. You can take this down. Right, so this is the main difference between the two. Whenever you come across the word chromatin or chromosomes, so you should know they're almost the same thing. Only thing, when it is present in more loose thread-like format, it's called chromatin. When it gets condensed, think of it like, a ball of wool, or a ball of thread, anything, whichever you're comfortable with. So if the yarn of wool or the ball of wool is, just stretched out, or you have thread everywhere, that single line of thread is what is the chromatin. Whereas if you make it into a ball kind of a form, that is chromosome. So it is still thread only, right? That's what I'm trying to put across. That it's the same material, it's the same thing, but it is existing in different shapes.
Miss, these other points like the cell and chromatin and all is part of chromatin, right?
This is all a
The
part of.
three big points.
Acha, three big points. Yeah,
Yes,
this
ma'am,
is
these three,
chromatin.
yeah.
This is chromatin. Yes, yes. Right. So chromosomes are basically, they are called the they are called the information or inheritance characters for DNA. Right. They carry the important information from one cell to the other when the cell divides. Right. So when the cell division happens, what happens is the chromosome from here is transferred to these two cells. So the chromosome is the one which contains the instructions and everything, and it will tell the cell what it has to become and what it has to do. Okay, basically you're just transferring your duties, or you're passing on your work to the next cell, something like that. So how is it passed on is by the chromosomes, and they are made up of substances which are called DNA and proteins. Okay, or protein, yeah, that's fine. DNA and protein. What is DNA? DNA is basically a unit of heredity which contains information. Right, so DNA contains the information for constructing and organizing cells. So what exactly happens is, when the cell divides, this cell doesn't know what it has to do. Right, we all come from one cell. We were all one cell at a point, isn't it? So how did that one cell, become your, maybe your nose, let's say your intestine, your skin, anything. You take anything, for example, everything is made up of cell only. Right. So how did that one cell become different parts in your body? How did it know that it had to become?
By replicating and by duplicating itself
Yes,
or
correct.
splitting it.
Yes, yes. So that by splitting itself, there were more cells, right? That's how you became a bigger in size. So you were not one cell. You became bigger, or you, as you are now. We are almost 4 to 5 feet. Let's just take an average number. Right, from one cell to become 4 to 5 feet, to achieve that amount of growth or size, the cell has divided. But how does the cell know what it has to do? Like, how does it know that it has to become the nose or intestine? Is by the nucleus. And why the nucleus is because it contains the chromosomes, which contain all these detailed instructions that, okay, you are supposed to go and become the nose, so your function is going to be so and so, right? Or you're going to become the intestine, so your role would be in absorbing nutrients. Or you're going to become the skin, your job is going to be protecting the other cells, becoming like a barrier. Okay, so this information the chromosomes contain. Right. Yeah, and this is about chromatin. So chromatin, basically, you just need to remember that the chromatin is the loosened version of chromosome, or the elongated version of chromosome. That's the only difference. It's the same thing. It also has DNA and protein, right? It is also responsible for passing on inheritance information to the next cell. It also does everything same. Only thing, when the cell is not dividing, there is, there is space for the chromosome to just exist peacefully, without existing in compact structures like this. So that's why it is existing in a loose format. Okay, right. Have you taken this down?
Almost, I'm writing chromatin.
Okay. Done?
I am done, but I think Rashmita and Jaydeep are still right.
Okay. Anybody still writing?
I'm done, right?
Okay.
Done, ma'am.
Done. Kushagra done?
Yes.
Aliza done? Okay. Roshmit, I think you're writing. Okay. Let me know once you're done. I think I missed somebody. I missed somebody. Yes. Neve, you're also done, right?
Yes, ma'am.
Okay.
I'm done.
Okay, great. Right. So these are some of the functions of the nucleus. Like by now you would have got the idea, right? When I say it is the brain of the cell. So here you just take down these words, just write function and just write cellular reproduction and write this as your second point. Okay, these are the keywords. Everything else is just explanation of the same thing. So just writing these underlined words would be sufficient for function. So basically, when you say cellular reproduction, like I said, because it contains the chromosome and it contains the information which needs to be given to the next cell. So the nucleus plays a very, very important role in cell division. So suppose the nucleus is damaged in a cell, right? Or if it is not able to perform its adequate functions. So cell division gets affected. And then when the cell divides, the recipient cells, or basically the two daughter cells which are formed, right, they will also get affected. Okay, that's what makes nucleus the control room or the main brain of the cell, right? It also plays a crucial part along with the environment in determining the way the cell will develop. Like I said, we all came from one cell, right? How does that one cell know that it had to become a huge organism?
They just split, right? They don't know. They just
They do know, right? The nucleus is the one which tells them that you need to perform this function. That's exactly what I'm trying to tell you, right? So the nucleus will tell the cell that you need to perform this function. The nucleus contains a set of instructions. You can think of it like instructions, right? So the nucleus contains these instructions, which it passes on to the cell. Okay, and then that cell develops into whatever type it needs to be. Like if it's going to become a part of the spinal cord or the brain, it has to turn into a neuron, right? So neuron has a different structure where it has axons and dendrites and things like that, right? So it transforms into a completely different structure and has parts and other accessories which will help it perform its function properly. So this job of telling the cell that okay, you become the neuron, or you become the skin cell, or you become the cell in your cheeks or anywhere, cell in your eyes, etc. That task is done by the nucleus. The cell just divides. Cell doesn't know what to do. The nucleus knows, right? Okay. Okay, done. Taken it down. Two words.
I'm just writing the last line.
Ma'am.
Yeah, you'll find some cartoons in most of the slides. Some of them are animated, but yeah, if I send the PPT, I think the animation gets lost. So yeah, these cartoons are just when you're revising or looking back at the PPT, you can remember that okay, this is this organelle or this is this part of the cell. Okay, done. Shall I go ahead?
Just 1 minute, please.
Okay. Okay, let's go ahead.
Yeah.
Right. So now that I had said that every cell contains cell membrane, cytoplasm, and nucleus. Right. This was what we confirmed previous class, and we did revise it now also. That every cell contains these three. But what happens in some cells? This nucleus exists as different forms. What do I mean by different form is, in some cells, the nucleus or the nuclear material is just scattered like this throughout the cell. Whereas in the previous figure which we saw, the nucleus was contained in a circular shape kind of a structure with a nucleolus inside, with a membrane, with pores, etc. All the other uh, accessories or whatever pores and other things it has, right? But in some cells, the nucleus does not have a predefined structure. So this is called a predefined structure, right? Or a fixed structure like this. It just floats randomly. So those kind of cells where they lack nuclear membrane and they lack this entire structure kind of a thing, they are called as prokaryotes. Okay, this is important. You can take these 3 points down. These are just extra information, the 3 subpoints, right? So in some cells, okay, this is not organisms, this is some cells, because nucleoid exists only in unicellular organisms. Unicellular organisms are just one cell length or one cell itself, right? So only in those, some of them, prokaryotes are found, where classic example is bacteria, some viruses, some fungi, etc. Right? Most bacteria are prokaryotes. Okay. So exactly what are prokaryotes is, basically they do not have this clearly defined structure, or they are poorly defined. That's what it's called, due to the absence of a nuclear membrane. Very important. You can use this as a point of difference between prokaryotes and eukaryotes. Okay, how did this word come into being, or what is prokaryote means? Prokaryote basically means false nucleus. Okay. And eukaryotes basically means true nucleus. Okay, U means true and pro means false here. Karyo is anything which is referring to nucleus and chromosomes. Okay, from there that word has come like that. Right, so basically prokaryotes are ones which do not have a clearly defined nuclear structure and they lack nuclear membranes. Okay, and organisms which have this clear structure and nuclear membrane are called eukaryotes. Right, remembering this much is sufficient. This is just some extra points that, if they don't really have a nuclear membrane, they lack other organelles which have membranes and things like that also. Means most of the things are scattered. They are not having proper structures like in the form of plant cells or animal cells that we are studying. Okay. So prokaryote example is bacteria. Eukaryote example you can take as any animal or plant. Okay. Or you can just say all multicellular. All multicellular will be prokaryotes.
Okay. We have to write this down, right?
Yes, yes. Whatever I tick mark, you can write that.
Okay, just 1 second.
Yes, yes. I'll wait. This is not required. Okay, this is example, right? Example is here, bacteria. Again, this diagram is not frequently asked. Sometimes they ask you to draw, sometimes. So just remember that you have to make the nuclear membrane scattered like here. Okay, just remember to make that. The other things are not important, that where is plasmid, what is ribosome, where is mesosome, all these things are not required. Just knowing that nuclear membrane is scattered like this is good enough. Sorry, nuclear material I meant, not the membrane. Right, so one or two differences are there here itself. The main difference between eukaryotes and prokaryotes is here itself, right? Presence and absence of nuclear membrane is your main difference. For other points, if you require 2-3 more points, you can refer to your chapter notes book. So at the end, if you see at the towards the end of cell chapter, there will be tables, okay, which have difference between different things, like there will be difference between unicellular, multicellular, prokaryotes, eukaryotes, etc. Right? So you can read that table for more points between eukaryotes and prokaryotes. Okay, Neve is done. What about the others?
Done, ma'am.
Okay.
I'm on the last point.
Okay.
Done.
Done? Okay. What about Aliza and Roshmit? Done?
Yes, ma'am.
Okay. Jaideep also done, right?
Yes, ma'am.
Okay, great. Right, and here is cytoplasm. So we finished cell membrane, then we finished nucleus, and we are left with the third feature which every cell has, which is basically the cytoplasm. So you can write all these 3 points. This point is usually asked as a give reason, hence it is included here. So it is important. Okay, so cytoplasm is basically the fluid content which is present inside the plasma membrane or the cell membrane. It's usually, if I say fluid, it's not exactly like water, but it's more of a jelly like or
Fluid, isn't fluid the mixture of liquid of liquid and gaseous state?
uh okay, yeah, it can be. It's not always necessary, but it can be. So here the cytoplasm, it's not exactly running, water like running consistency. It's more like a jelly like, or if you would have seen, I don't know, do you all know slime? Have you seen slime before?
Yeah, you can just say semi-liquid. I mean semi-solid, right?
No, I wouldn't say semi-solid. If I say semi-solid, then the organelles would not be able to, stay inside it. They would be fixed in one place. But everything floats around the cell, so it is not semi-solid. I would say liquid or fluid, you can call it that. And it's more of like a slime like, this is for your information that it is slimy. It's not, it's not going to flow. If you cut open a cell by chance, it's not like blood. It's not going to come gushing out. Blood also is a little bit thicker than water, right? It does not really flow off like water. This is even more thicker. It's more slimy. And I think the perfect adjective is slimy. I can't think of anything better, right? Slimy, jelly like, something like that. I have a video towards the end. I'll show you. In that video, there is the cytoplasm
Okay.
and all organelles, everything. So I'll show you why am I saying slimy like. Okay. Right. So it becomes the basis where it holds all the cell organelles, everything, nucleus, every organelle which is there inside that cell is placed in the cytoplasm. It's basically floating there. You can think of like if you go in a pool, all the the lifeboat kind of thing, pool noodles, everything are just floating in the pool, right? The same thing. So everything is floating in this cytoplasm in a cell. Okay, this is an important give reason. Why? Because it talks about the significance of membranes. Why are membranes important? Okay, why do every cell, or why do we say that every cell has membranes, or what is the importance of membranes? Is basically that you could see that with the example of viruses. Okay, so now viruses are unique. Why? Because they exist. Okay, they are basically not substances, I would say organisms, which are existing on the borderline of non-living and living substances. Okay, why do we say this? This is exactly explained here. Why do we say this? Is because virus, when it exists on its own, it does not show any characteristic of life. And why is that? Because it does not have any membrane. Okay, so like we said, every cell has a cytoplasm, every cell has cell membrane, and every cell has nucleus. So because this lacks membrane, we say that it is not living. But when the virus attaches itself to a host, or basically any other living body, it gets the membrane from that. It takes over everything from that cell. Okay, so it gets membranes, everything, and it becomes a living substance. Okay, yeah. So from here you can use this point also when we say that every cell has three substances. Then why do we say that viruses are not living, or why do we say that they are on the borderline? Is because they lack membranes, and only when they,
And they can only activate when they get inside a user in a body, living body.
yes, correct.
Let me just give me 5 minutes to read this, please.
Okay, yeah, take this down. Right, when you're writing this point, you can write give reason next to it, or you can just put a small star next to it, right? It's important. Done?
Yes, ma'am.
Okay,
Done, ma'am.
done. Okay. Okay, three are done. What about the rest of you? Done?
Done.
Yeah,
Done. Anvesha, Alisa, done?
Oh yeah.
ठीक है, okay. Right. Now that we finished features of every cell, we finally come to the big part or the major chunk of our chapter, which is the cell organelles. Okay. Again, you don't need to take this down. We are going to be doing all of these only, right? We go one by one. This is all, you will see that the PPT is being made in such a way that it helps you while you're revising also. So this all will come in handy when you're revising, that you can just look at this chart and just check that, okay, have you covered these topics? Then you're good to go, right? Okay. So these are the main 7 organelles we would be covering. So we've already covered nucleus, right? And we will be doing endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. Of course, the cell is not limited to only these organelles. There are many more things floating inside the cell. But at this stage, these are the essential ones which you need to know. Okay, the basic ones or which are required to know would be these 7. Okay, right. So we first will start with endoplasmic reticulum. So you can take this down and you can take this point down. The second point is not required. You can just put round or oblong bags here itself. Right. Okay. Diagram not required. We will be drawing the animal cell and plant cell diagram, right, at towards the end. If we get time, we'll draw, or I might ask you to draw it on your own. So this all the organelles would be covered in that, so we won't be drawing them separately. Okay. So basically, endoplasmic reticulum, where is it found? It is usually attached to the nucleus. Okay, you can take that also down. It's attached to nucleus. Okay, and it's a large network of membrane bound tubes and sheets. Okay, I don't know. Are you all Harry Potter fans by any chance? Any of you watched Harry Potter?
Yes, ma'am.
Yes, yes. Okay,
I watched the movie.
watch the movie. Okay, right. So in Harry Potter, if you would have seen the Goblet of Fire movie, there is one maze, right? One maze which they have to go into. So think of it. Think of the endoplasmic reticulum just like that maze. Okay, there is a huge maze kind of a thing. Okay, which extends all the way. Yes, Sahil, you were saying something. I'm sorry, I couldn't hear you.
No.
Okay. 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 So this huge maze or this huge pathway stretches all the way from the nucleus towards the end of the cell. Okay, it usually spans the entire cell. Okay, it is there like this towards the end of the cell. So what happens is whatever needs to move through, that we'll see in the next slide, that it's usually proteins. Proteins are manufactured here, and then these proteins, whenever they need to travel to other parts of the cell, they make use of this tubes to travel, and wherever they need to exit, they'll just move on. So the main function of an endoplasmic reticulum is basically it's a pathway, just a huge maze. Right, and then there are two types. One is called the rough endoplasmic reticulum, and one is called a smooth endoplasmic reticulum. Again, we will discuss this in detail in the next slide. What are these?
Okay, just give me 2 minutes.
Okay, yeah. So you just need to take down the first and the third point. That's sufficient. No diagram required.
Done, ma'am.
Done?
No, almost.
Okay. Others done?
Yes, ma'am.
Yes, ma'am.
Okay, I'll go ahead. You can write those points on the next slide. It's fine. Right. So we said that there were two types. So one is the rough endoplasmic reticulum and one is the smooth endoplasmic reticulum. Now, why is it called rough? Is because it has ribosomes attached to its surface. Again, you can take down all the points. Usually from this slide onwards, I think all the slides and all the points would be important, right? Because they just have the structure, function, or location kind of information given. Okay, so rough endoplasmic reticulum is called rough or it has a rough texture. Why? Because there are these small dot-like structures on the surface. Think of it like that, right? When you have a boil on your skin. Okay, suppose you have a heat boil or just a boil, temporary boil on your skin. That part of your skin becomes rough, right? Whereas if once it goes away, it becomes smooth. It's back to its smooth texture. So think of it something like that, where in the endoplasmic reticulum, if this is your maze like channel like thing, right? So this has dots or ribosomes attached to the surface. And what do these ribosomes do? That these ribosomes are the protein manufacturing sites. Okay, very important. It's usually asked as a one marker question or fill in the blank question, something like that. Dash are the protein manufacturing sites. So it is ribosomes. Okay. And this, and what is the function of the rough endoplasmic reticulum then? This rough endoplasmic reticulum transports this protein which these ribosomes make to various parts of the cell. Like I said, wherever the protein needs to reach. Suppose it needs to exit here. So it will exit here and it will find its way to the Golgi apparatus, etc. and go ahead. Okay. So the main job of endoplasmic reticulum is it acts as channel transport. Okay. The functions of endoplasmic reticulum is there again in the further slide. So I'll tell you where to put the heading function and write that. Okay. Oh. Okay. Now what happens to smooth endoplasmic reticulum? Smooth endoplasmic reticulum is usually the same part of this channel. Okay, this is the same extension of this transport channel or this maze, you can say, right? But the only thing is the smooth endoplasmic reticulum, you can take a guess. If I say it is smooth, then what would it be like? Okay, if I say the rough endoplasmic reticulum is rough because ribosomes are attached to it. Then if I say smooth endoplasmic reticulum will be smooth because
And because there are no ribosomes attached to it.
yes, excellent. Right. So if I'm telling you the rough texture is because of the ribosome, then the smooth texture will be absence of ribosomes, right? So it does not have anything attached to it. Absence of ribosome. Okay. This I'm just showing you because I made the figure here. We have SER in the next slide.
Okay.
To be written. Okay, I'm just explaining the difference because right now you've not seen anything, you don't know what I mean, all this must be new, right? So it's difficult to comprehend that where exactly it is and what exactly does it do. So I'm making these rough diagrams so that you can visualize a little bit where it is. Okay, so SER is also an extension of this same endoplasmic reticulum. Only thing is the rough endoplasmic reticulum is usually towards the nucleus, closer to the nucleus. Smooth endoplasmic reticulum is closer towards the cytoplasm. Okay. And they don't have ribosomes. So ribosomes, apart from the RER, so apart from being attached to this RER, ribosomes also float in the cell. Okay, they are also attached or they are also found as free flowing structures in the cell like this. Okay, there also their job is the same. They will be manufacturing protein only. Okay, right. Okay, next we come to smooth endoplasmic reticulum. This is important. Again, everything important, you can take it down. So smooth endoplasmic reticulum has a little bit different function compared to the rough endoplasmic reticulum. Okay, it basically helps in the manufacture of fat molecules. So in the first class when we discussed about plasma membrane, I had told you, right? Plasma membrane or cell membrane is made up of a type of fat molecule which is called lipids. So this lipid is made here in the smooth ER or SER. So this is exactly the second point. So that lipid which is manufactured in the SER is used to make cell membranes. Okay, and the process of making the cell membrane is called membrane biogenesis. Okay, biogenesis, if you break the word down, bio, of course, you know, right? It refers to any living substance or any living thing. Genesis usually means creation. So when I say biogenesis, membrane biogenesis, I'm saying creation of membranes. Okay, so the lipid which is made in the smooth ER is used to make this right here. You can see, right? This is your cell membrane or your plasma membrane. So the lipid is used to make the cell membranes, right? Also, there are some other functions also. Not everything is used to make this. Some of it which is extra, or some other types of proteins and lipids which are formed, they function as enzymes and hormones for different purposes. And this is also important. This is an important point. It usually is asked as a fill in the blank type question, where they'll ask you that dash detoxifies many poison and drugs. Basically dash detoxifies poisons. So you would have to say liver cell of vertebrates. Very important. The whole keyword becomes this entire phrase. Right? Liver cells of vertebrates. And in that, the SER present in the liver cells of the vertebrates, that helps in detoxifying poisons and drugs. Okay. What are vertebrates? Basically, they are organisms which have a spinal cord. In case you are not aware, you can take it down. Organisms which have spinal cord. Basically, any organism which is able to stand upright. Right? Animals, humans, we are all vertebrates. Whereas worms, eels, some kinds of fishes, snakes, they can just glide around, right? They are not vertebrates. They don't have a spinal cord, right? So all of this is important. They might ask you what.
This is used to make.
Yeah, I'll wait. They might ask you a question as what is membrane biogenesis, right? So your answer would be formation of cell membrane. Building of cell membrane is called membrane biogenesis. Done?
Just a minute, ma'am.
Okay.
Done, ma'am.
Okay. Done, right? Shall we go ahead? Okay. Okay, Neil. Right. Yes. Next we come to the functions. Right. So basic function again, like I kept reiterating. Main job would be it acts as transport channel. Right. Sorry. You can take down the first point as it is. Second point, you can just write it acts as a cytoplasmic framework. Okay. So these are the functions mainly, yeah, like I discussed. So what is cytoplasmic framework I want to highlight here? So suppose this is your nucleus and your endoplasmic reticulum stretches, like I said, it stretches from the nucleus to more than half of the length of the cell, right? So because of this, what happens is it gives the cell some kind of a framework. So like in plant cells, you have your cytoplasm, sorry, your cell wall and the cell contents giving it some kind of framework, right? Because of the wall pressure and turgor pressure. Additional help is provided by the endoplasmic reticulum in maintaining that kind of a structure. And also what happens is because it spans this much length, they are saying biochemical activities, right? Biochemical activities basically refer to some kinds of reaction where uh, some reaction is occurring and some type of a biological substance is being produced. Okay, like you might, we might come across in brief in mitochondria, right? That some reactions occur and ATP is produced, which is basically energy. So this surface, all this length, this becomes like a surface for these reactions to occur. So that's what it basically means. It becomes a sort of like a skeleton, and it also provides space for these reactions to occur. Oh, sorry, sir. Yeah. Yes, Neem. You had a question?
No, ma'am.
Achha. Right. So wherever you need to read the difference between, or wherever you can look at the difference between at that point, so you can refer to your chapter notes. These tables are there in the chapter notes, which is why I did not include it here. Right, where if anything was missing there, then I've included it here. Okay, you can read the difference between smooth and rough. Again, you already have 2-3 points, right? You can say that rough has ribosomes. While doing it only, we came across 2-3 points. Then smooth will not have ribosomes. Then you can state their functions as a difference because rough is majorly involved in transport. Smooth, apart from transport, is also involved in the manufacture of fats and some types of proteins, right? Fats in the form of lipids and some types of proteins. So that becomes your another point of difference. Then another point you could state would be rough is closer to the nucleus, smooth is a little bit further away, right? Like I mentioned. So you already have 2-3 points. For some additional points, you can refer to that table there. It would be having some more specific points given. Okay, right. Done? Shall we go ahead? Done. Okay. I think Roshmit is writing. Sahil, are you done?
Yes.
Done, ma'am.
Okay, great. Right, now we look at the next organelle. Yes, which is the Golgi apparatus. You can pronounce it as Golgi, Golgi, anything. Okay, whatever pronunciation is fine, that does not matter. But make sure you're spelling it correctly. Okay, Golgi apparatus, Golgi. It's also called Golgi body, or it's also called Golgi complex. So if you come across any of these terms, they all refer to the same organelle. They are not different. Okay, they are the one and the same. So just if you want, you can make a note of this as well, right? That all these three terms refer to the same cell organelle, which is the Golgi apparatus. And here you need to know the scientist's name also. So the scientist who discovered and described the structure of this organelle was known as Camillo Golgi. Right. And what is this? So this is something a little bit about their structure, which is it consists of a system of membrane-bound vesicles, right? Attached. You can just write this. Okay, take down the whole thing. That's fine. Even if you ignore the arranged approximately parallel part is fine, right? And this is not required. First and second point is only required. Third point you can skip. Okay, once you are, let me know once you've copied the first 2 points, because then I would like to explain what exactly this cistern means, what exactly vesicle means, and what is the function of Golgi apparatus. So after you finish writing, I'd like to discuss that because that is something new, so you would have to pay attention to the screen for it, right? Okay. Done? Taken it down?
I'm done.
Okay, great.
Done.
Done. Okay. Right. Yeah. I waited because I wanted to explain these 2 points. Right. Okay. So now to understand what vesicles are, we need to understand how substances enter into a cell or how do they move around the cell. Okay. It's not like it's going to just enter and whatever it is, the salt, or you're talking of some water molecules, or you're talking of nutrients, anything. Like how do they exactly move around in and about the cell? Even if you're talking about proteins that we said, right? The proteins are going to move from endoplasmic reticulum to wherever the cell requires the protein. So how exactly do they move? So what happens is they form something which is called as a vesicle. So suppose, take for example, any substance entering the cell. Okay, suppose water is entering the cell. So what happens is there is a small cavity which is formed like this. Okay, and then the cell gets, so this is your membrane, right? So, and if this is, okay, yeah, okay. So this is the place where the water molecule has entered. Suppose this is your water molecule, right? So what happens is this part gets pinched off like this and you will be left with a bubble and your water molecule inside it. Okay. And this bubble is basically what is called a vesicle. Okay, so this bubble kind of a structure is called a vesicle. So what happens is even when the protein is moving out from the endoplasmic reticulum, okay, if this is our endoplasmic reticulum and the protein is going to move out from here, let's say, okay, same color over here, right. Yeah, if the protein wants to move out from here, let's say, so what happens is there will be a bubble like shape formed, and inside that bubble the protein is going to move around from wherever it came out to wherever it wants to go. Okay, so when it reaches the Golgi apparatus, this bubble along with the protein will enter the Golgi apparatus and will absorb into the Golgi apparatus. So this is how it works, and this bubble kind of a structure is what is called as a vesicle. Okay, membrane bound means, yeah, it has a covering kind of a structure, that is your membrane, and inside that whatever material is being transported or whatever is being moved is present inside that, so that it is securely or safely transported from wherever it came from and wherever it has to go. Okay, right. So that is vesicles. And another thing what I wanted to tell you was what are cisterns or why are they called cisterns? So if you see the diagram here, right, these flattened sac-like structures, these things, these are what are called as cisterns. Now, where did that name cistern come is because cistern usually refers to any container where you're holding water. Basically you're holding something or you're storing something. Okay, so cistern is basically a unit for storing water. And what happens in the Golgi apparatus is once it takes in these protein vesicles, it stores them here. That is one of the main functions of Golgi body or Golgi apparatus, is storage. Right, so because they store these proteins which are in the form of vesicles and they have some amount of fluid, etc. in them, hence that name cisterns came into being. Right, so you can just connect the two like that, that okay, cistern basically means it is storing or it is holding something, and what is that something is basically the proteins. Right, so this point you can ignore for now. They're just saying that okay, they are connected with the ER. Why? Because the protein which is coming out from the ER has to eventually enter the Golgi body. So they are always closer to the ER itself, endoplasmic reticulum, because the proteins from there can easily then enter the Golgi body. Right. And why is that is there in our next slide. That, yes, the functions, right? So the main function of the Golgi body or the Golgi apparatus is storage, modification and packaging of products in vesicles, right? Second thing is formation of lysosomes also. And in some cases, complex sugars may be made from simple sugars, right? Basically, simple sugars are molecules like sucrose, fructose, etc. And complex sugars are glucose, etc. Right? Just I'm giving you an example, not required to know what are simple complex sugars. Main job you need to know would be the first point and second point. Lysosome is another type of a cell organelle, which we will study in a bit, right? In the maybe the next slide or so, right? Okay.
I'm gonna just give you 5 minutes. I'll just write this down and go to the washroom.
Yeah, I'm waiting. Okay. So what happens basically is, if this is your nucleus, right? And you have your endoplasmic reticulum like this. Okay. Then the Golgi body will be somewhere here. Okay. This is what it looks like, right? And suppose this is your protein, which is being manufactured in the ribosomes, right? Okay, let this be the ribosome. Let this be the protein. So now that the protein is coming out in the form of a vesicle, right? And you have your protein inside here, right? This enters the Golgi body where it is packaged and stored. Packed and stored. Stored till when? Till the cell, requires it for some function, or whatever it has been called that okay, you are required to perform this kind of a function. Till that time it is stored in the Golgi apparatus itself. Again, here you can take the example of when you are going to a restaurant and you are ordering something for takeaway. Let's say you order something for takeaway for anything, McD, you go to McD or any restaurant and you order for takeaway. What do? They'll first prepare your food. Okay, I don't like yellow color. Can't see anything. Okay, pink is nice. Okay, right. I'll write here. So when you order takeaway, what happens is first they prepare your food, right? Whatever you ordered. Suppose you ordered a burger and let's say some fries or something like that. So first they'll prepare your food, right? Then they don't just give you the burger like that without putting it in anything, without covering. They just don't hand you the burger and be like, you managed to take it, right? They don't do that. So the same way the food is prepared in the ER, right? And the next step would be they would be packing it for you. They'll put it in a nice box, then they'll add some paper bags or a plastic bag or some kind of a bag, right? And then give it to you. So this packaging is done in the Golgi body, right? So this is the procedure which happens. Then the protein which finally is packed or nicely packed and stored and given a beautiful gift wrap kind of a thing, then that protein is used for performing whatever cell functions it needs to do. Then it comes out of the Golgi body and moves to wherever location is required, wherever it is being called, right? Okay, let me know once you are done with this. I want to show you one small video where the vesicles and everything is there explained nicely. Okay, Neve is done. Okay, Roshnit is also done.
Done.
Done. Okay. Done. Everybody done? Okay. Okay, I'll quickly show you that video then. Right. I thought we might be able to complete the chapter today, but it doesn't look like it. No worries. I will take one more class to finish then. Right. This animation is not working in full screen. I have no idea why. I'll show you this also. Right. See, this is how it goes. So you can see the proteins going inside, right? So once they go inside like this, they get packed and then they come out. Now, like that, the animation should be there for the others also, which means it was not moving there also. Let me go back. Oh, this is just not here. Cytoplasm is also animated. It moves like this, right? Yeah, jiggly jelly kind of thing. Right, the slime is a classic example. That's why I said even in slime, if you play with slime, whatever substance you add to it, it just gets stuck on it, right? That's exactly what happens in the cytoplasm. Okay, I want to show you a video. Okay, yeah, then we do the others next time.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. Inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts.
Okay, yeah, you saw the jelly-like thing, right? So that's how they were floating inside the
I
cell.
must have seen this video in school, so we know it's like.
Oh, you've seen this video in school? Great.
Yeah, I remember it.
Oh, okay. Others, have you all also seen or have you?
Yes, ma'am.
And I was not able to hear. Oh, I think I have some audio issue. Wait, I'm not. Ah, I think I did something wrong with my internet. Just give me 1 second. Acha. Okay, and you said you have also seen this video in school. Okay. You've seen it. Great then. Yeah, then understanding what is vesicle would be easier, right? Since you've seen it. Okay, great. And it's not working here. Okay, sad. Yeah, my screen frozen only. I don't know what happened. Okay. Okay, since you have seen this video, then okay, great. This is a good one. I was not able to find any other video which was as good as this. Next we are left with lysosomes and
Back
arcuoles, mitochondria, and cell division.
Chloroplastism.
Yes, sorry. I'm having some issue, I think, audio issues.
Oh, sorry, wait, I was on mute. Chloroplast is in the plant cell, right?
Yes, chloroplast is in plants. Yes, it's in plants. Okay, yeah. So we have 4 more organelles to cover. So which, of course, it's not going to be possible in today's class. So I will need another class to complete the chapter. Let me see then if I can search for some other video, I'll do that, right? And then I wanted to show you this. There is this very nice animation, kind of a simulator. Okay, please work, right? Okay. Once we finish the chapter, no, I'll share this link with you. Here you can keep playing around with different organelles. You can identify them. See, they'll give you the, they'll give you the name. You have to figure out from here which is the vesicle, which is the ribosome, which is the etc. Right? Centriole is not required for you, but the other things like, where is the endoplasmic reticulum, where is Golgi apparatus, mitochondria, and all that. And even animal cell is there and plant cell is also there. If you see here, if you go back here, now plant is there. Yeah, here. So here also you can play and identify. This we'll do next class once we finish the remaining 4 organelles which we are yet to cover, right? So once I finish all that, I'll share this with you so you can play around with this, and then it will be easier for you to remember the diagram, right? That okay, this organelles are there in plant cells, these are there in both. Okay, right. So then for today's class, you can submit your notes once the class ends. And yes, before our next class, please make sure you revise whatever we did today and then come. Okay, okay. See you all next class. Bye bye.
Thank you, ma'am. Bye.
Bye, ma'am.
Bye, ma'am.
Thank you, ma'am.
Bye.
Bye.
Thank you, ma'am. Bye, ma'am.